This is my 11th video in my AP Biology review series, and it is about the chromosomal basis of inheritance. The chromosome theory of inheritance links inheritance with the behavior of chromosomes during meiosis. It states that Mendelian genes have specific loci, remember loci are the location of the genes, on chromosomes, and it is chromosomes that undergo segregation and independent assortment. So you might remember in my last video, I talked about how Mendel discovered the laws of segregation and independent assortment, but he discovered that in regard to genotype and alleles, but he didn't know how that tied in with chromosomes. So the chromosome theory of inheritance kind of links the two together. So as you can see in the picture, we have meiosis occurring, and it is the independent assortment of the chromosomes and the segregation of the chromosomes that leads to the patterns that we noticed in the separation of the alleles and the resulting phenotypes and genotypes. Thomas Morgan lived from 1866 to 1945, and he experimented with fruit flies to better understand genetics. Wild type is the normal phenotype. It's red eyes for the flies that he experimented with. Specifically, the flies are called Drosophila melangister, but we're just going to refer to them as fruit flies. And we're going to represent that as a W with a plus because of the mutant phenotypes, which are the alternative phenotypes. It's white eyes in the flies, so that's why we have a W, because the mutant is white eyes. Morgan mated a wild type female with a mutant male. The F1 generation all had red eyes. The F2 generation, sorry, I mentioned in my last video, but I didn't hear yet. F stands for filial, so that's F1 generation is the first filial generation. It's the first kind of children of the parent generation, and then the F2 is the children of those of that generation. The F2 generation had a 3-to-1 ratio of red eyes to white eyes, but he noticed that no females had white eyes. He knew that white eyes was a recessive trait because all of the F1 progeny had red eyes, none of them had white eyes. And he hypothesized that the eye color gene was only on the X chromosome, and that there was no corresponding locus on the Y chromosome because all of the offspring that had white eyes were all male. So because of that experiment, he realized that genes located on a sex chromosome show unique inheritance patterns and don't necessarily follow the Mendelian patterns. Linked genes are genes located on the same chromosome, located close to each other, that are inherited together. Another experiment that Morgan did was mating wild-type flies that had gray bodies and normal-sized wings with mutant flies that had black bodies and vestigial, which are small wings. He found a much higher percentage of parental phenotypes than he expected. Parental types are offspring that inherit a phenotype that one of the parents has. Recombinants are offspring that have non-parental phenotypes. If 50% of offspring are recombinants, there's a 50% frequency of recombination. Two genes on different chromosomes have a 50% frequency of recombination. In Morgan's experiment that we mentioned in the last slide, with black normal-sized flies and gray vestigial flies, he noticed a 17% recombination. Again, that's much lower than the 50% that we expect, so that means that the two genes must be linked. They must be inherited together. Recombination frequency is connected to the distance between linked genes. Because if you think about it, if the two genes are very far apart on the same chromosome, even though they're on the same chromosome, they're probably not likely to be inherited together. So the closer they are together, the lower percent of recombination, because it's very likely that they will be inherited together. Here's an example. We talked a lot about peas in the last video. Here's an example which shows what happens if they're not linked. We have a parent that it has yellow pea color and a round shape, but is heterozygous for both of those traits, and then another parent that's homozygous recessive for both of those traits. And as you can see, 50% of the offspring have parent types. They have the same phenotypes as the parent, and 50% have different phenotypes. Those are the recombinant types. So this is what happens if the two genes are not linked, if they're inherited independently. A genetic map is a list of gene locations along a chromosome in order. A linkage map is a genetic map based on recombination frequencies. The further apart the genes are, the greater the recombination frequency. Again, because if they're close together, there's a high chance that they will be inherited together. Map units are the distances between genes. One map unit corresponds to 1% recombination frequency. So again, as you can see, the closer apart they are, the lower the recombination frequency. The further apart they are, the greater the recombination frequency. If two genes are more than 50 map units apart, they can be physically linked because they're on the same chromosome, but are genetically unlinked because they will assort independently as if they were on different chromosomes because they're so far apart. So again, as we said in the last slide, if the recombination frequency is 50% or higher, they're not linked. If it's lower than 50%, they're probably linked. Let's do an example with the map. If genes A to B are 12 map units apart, B to C is 8 map units, and A to C is 4 map units, what is the order of the genes? So it's pretty easy. We have two options. This is one of them. We have A, C, and then B. We know that C must be in between them because A to B is 12 and B to C is 8 and A to C is 4. So 4 plus 8 adds up to 12 from A to B. And here is that same thing flipped. It could also be B, C, A. So if you look at these pictures, A to C have a lower recombination frequency because they're closer to each other, and then A to B would be higher because they're further away. But they're all still linked because, again, we're under 50. Sex-linked traits. An example is color blindness. It's on the X chromosome. It's sex-linked recessive. Female heterozygotes don't express the phenotype. Any male that inherits a recessive allele from the mother will exhibit the trait because, keep in mind, males only have one X chromosome. Another example is hemophilia. It's a sex-linked recessive disorder, again on the X chromosome. Proteins needed for blood clotting are absent, and clots are slow to form. So, unfortunately, affected individuals will need to receive injections in the missing protein in order to supply their blood with the factors that it's missing. So on this side we have a pedigree we talked about in the last video too. And let's do this example together. Which of these individuals must be carriers? Remember, carriers for sex-linked disorders must be female because it's an X-linked disorder. So we know that this individual must be a carrier because she has a son that has the disorder, but her husband and her husband doesn't. But the fact that her husband doesn't really matter, because the son inherited the X chromosome from the mother. We also know this individual must be affected because, again, she has a son who's affected. And again, we know that those two individuals that I marked with X's, they're not actually affected. Sorry, I accidentally said that for the second person. They're carriers, right? Because if they were affected, they would be completely shaded in, but they're not. They're carriers. And we know that that individual must be a carrier because they have a child that is affected. And these are the three people we definitely know are carriers. There could be another few that have a chance of being a carrier, but we're not 100% sure because they don't have children that are affected. Chromosome mutations. Non-disjunction is when a pair of homologous chromosomes do not separate in meiosis 1 or meiosis 2. This leads to aneuploidy, which is an abnormal number of a specific chromosome. And unfortunately, this leads to many problems in the offspring. Trisomy is 3 of a particular chromosome. Monosomy is 1 of a particular chromosome. Polyploidy is having an extra complete set of chromosomes, which is actually pretty common in plants, surprisingly. So this is where this picture shows meiosis, but the non-disjunction occurs in meiosis 1. But in this case, it occurs in meiosis 2. So as you can see, the non-disjunction leads to trisomies, a monosomy. It really messes up the amount of chromosomes in each cell, unfortunately. Alterations of chromosome structure. There's deletion, where a segment of a chromosome is removed. Duplication, where a segment of a chromosome is repeated. Inversion is where it's reversed. And translocation is when a segment of one chromosome is moved to another non-homologous chromosome. So this picture shows all of them in order. The first one, as you can see, is a deletion. We're removing the gene C. The second one's a duplication. We have that same gene copied over twice. Inversion, flipped. And then the last one shows a translocation, where we're moving a piece of a chromosome to a place where it doesn't belong. And unfortunately, all of these will lead to issues in the offspring. Extranuclear inheritance. Some genes are located in organelles in the cytoplasm. Remember that mitochondria and chloroplasts each have their own DNA. They will reproduce and separate into daughter cells without following the Mendelian rules of inheritance. In most plants and animals, mitochondrial DNA is inherited maternally from the mother because almost all of the mitochondria passed on to the zygote came from the cytoplasm of the egg cell. So it's really rare for people to be heterozygous for mitochondrial genes because they're inheriting it all from one parent. These genes, not surprisingly, will code for proteins that are important in the electron transport chain and ATP synthesis because that's what the mitochondria's main job is. So here's a picture of the mitochondria. As you can see, there is mitochondrial DNA in it that's separate from your chromosomes, but is still inherited. That's it for this video. Thank you so much for watching, and please subscribe if you would like to see more videos.